IB Bio, Animal Physiology, Topic 11, Part 8, Kidney Osmoregulation C, focuses on the role of the loop of Henle, the role of the collecting duct, and the hormone ADH in regulating water loss in urine, as well as excreting high concentrations of urea in the urine. Secondly, this movie will look at how we diagnose and treat kidney disease. The essential idea is all animals excrete nitrogenous waste products, and some animals also balance water and solute concentration. Here is the outline of all the available movies for Topic 11, Animal Physiology. Use this outline to choose the movie you need for review. This movie is focused here. Here's an IB syllabus statement that I presented in Part 7, Kidney Osmoregulation B. I will open this movie by providing you the opportunity to review this material. Annotate diagrams of the nephron, including the glomerulus, Bowman's capsule, proximal convoluted tubule, loop of Henle, the distal convoluted tubule, and the relationship of the nephron to the collecting duct. Here is a nephron, colored yellow. The glomerulus is a ball of capillaries enclosed by Bowman's capsule. Ultrafiltration occurs in the glomerulus, and the filtrate passes through the proximal convoluted tubule to the loop of Henle that descends into the medulla of the kidney. The filtrate passes up the ascending limb of the loop of Henle back to the cortex region of the kidney. Then the filtrate passes through the distal convoluted tubule and then leaves the nephron for the collecting duct, which is not shown here. Here's a chance to practice the annotation of a nephron. With the exception of number 6, which is a small vein known as a venule, you should be able to name all the other structures. Turn the movie off now for practice. Here are the named structures. Spend some time with this slide. Turn the movie off again. Notice the parallel nature of the descending limb of the loop of Henle, the ascending limb of the loop, and the collecting duct. The IB syllabus wants you to notice the close relationship of the nephron to the collecting duct. Look carefully at this diagram to notice the three processes occurring through the various regions of the nephron and the collecting duct. Now keep in mind that these capillaries are always in close proximity to the tubules of the nephron and the collecting duct. We have ultrafiltration in the glomerulus and Bowman's capsule. We have reabsorption of glucose, sodium, and water in the proximal convoluted tubule. We have secretion of ammonia and hydrogen ions in the distal convoluted tubule. And it's important to note that while most of the organic molecules are actively transported across cell membranes, water moves by osmosis. Ultrafiltration at the glomerulus prevents large proteins from entering the filtrate. And while glucose does pass from the blood to the filtrate, nearly all of it, except for a little bit used in cell respiration, is returned to the blood by reabsorption. The urine should have no glucose. The reabsorption of glucose from filtrate to blood occurs in the proximal convoluted tubule. Once again, here is a good slide to study. You can see the close relationship of the capillaries to the tubules of the nephron, and the parallel nature of the descending limb of the loop of Henle and the ascending limb of the loop of Henle and the collecting duct are important to the next part of our story, which focuses on water regulation and the excretion of a urine concentrated with urea. Here are the IB syllabus statements that drive this movie forward. Explain the roles of the loop of Henle, medulla, collecting duct, and ADH in producing a concentrated urine. State that the loop of Henle maintains hypertonic conditions in the medulla, and state that ADH controls the reabsorption of water in the collecting duct. I've come back to a simple image of the nephron, one you've seen before, to note the parallel relationship of the descending limb of the loop of Henle, the ascending limb of the loop, and the collecting duct. The counter current flow of the fluids in these three tubules serves the larger process of conserving water osmoregulation, as well as excretion of a urine concentrated with urea. Remember, the loop of Henle and the collecting duct descend into the medulla of the kidney. The glomerulus, Bowman's capsule, the proximal convoluted tubule, and the distal convoluted tubule are located in the cortex of the kidney, while the loop of Henle and the collecting duct descend into the medulla. The filtrate enters the loop of Henle in the cortex, descends into the medulla, then returns to the cortex before descending once and for all in the collecting duct toward the pelvis through the medulla. Once in the pelvis, the urine moves to the bladder in the ureter. Again, filtrate enters the loop of Henle in the cortex. The filtrate moves within the descending limb of the loop of Henle into the medulla. Then the filtrate moves up the ascending limb of the loop of Henle back into the cortex. Then the filtrate moves into the distal convoluted tubule before entering the collecting duct, where it traverses the medulla from the outer edge of the medulla to the interior of the medulla before being released into the pelvis as urine. In moving to the next slide, I want to add that the details of nephron functioning are highly complex, more complex than I can explain at this level. I will present a simplified explanation of the roles of the loop of Henle, the medulla, and the collecting duct in producing a concentrated urine. Be patient, pay close attention, and feel free to ask questions in class. In this diagram, you can immediately see the detail on the movement of materials in and out of the nephron tubules. You can see the concentration of the filtrate given in milliosmoles per liter. Notice how the concentration of the filtrate increases in the descending limb of the loop of Henle, and then decreases in the ascending limb, and then increases once again in the collecting duct before leaving the kidney as urine. This is the concentrated urine that you excrete. One other thing to notice before I start in on an explanation, the light coloration of the diagram in the cortex indicates that the interstitial fluid within the kidney, surrounding all of the tubules in the cortex, is relatively dilute, hypotonic. The dark coloration in the medulla indicates that the interstitial fluid surrounding the tubules is relatively concentrated, hypertonic. The concentration of the fluids surrounding the tubules displays a gradient from low concentration in the cortex to high concentration in the medulla. The loop of Henle establishes this gradient, and then the filtrate passes down through the collecting duct, which spans this gradient. Let me say that again. The loop of Henle establishes this gradient, and then the filtrate passes down through the collecting duct, which spans this gradient. The concentration gradient in the interstitial fluid right here serves to draw water out of the collecting duct as the filtrate moves through the collecting duct. Thus, water is conserved, returned to the blood, and the urea is concentrated in the urine. Be patient as I work toward the simplified explanation of these events. In this diagram, you can see that the filtrate increases in concentration in the descending limb of the loop of Henle, then decreases in concentration in the ascending limb of the loop of Henle as it moves back up to the cortex. Now, this is where things get really interesting. The dilute filtrate enters the collecting duct in the cortex and begins its descent through the medulla. The interstitial fluid surrounding the collecting duct is increasingly more hypertonic, serving to draw water out of the collecting duct to be taken up, conserved by the blood. The urine, as it exits the collecting duct into the pelvis of the kidney, is very, very concentrated with urea. Here's the simplified explanation of how the loop of Henle establishes the concentration gradient from the cortex to the medulla, the gradient through which the collecting duct must pass. The gradient is seen in this image as a color change from light brown in the cortex to dark brown in the medulla. Now, the process is far more complex than my simplified explanation, involving a countercurrent flow that you can see here, but as a way to understand what's going on, let me start here, deep within the medulla, with the filtrate highly concentrated at the bottom of the loop of Henle. Now, the filtrate is highly concentrated. I will come back to how that's occurred, but as the filtrate moves up the ascending limb, it's important to say that the ascending limb of the loop of Henle is impermeable to water, but that salts can passively, then actively be moved out of the tubule. And this passive and then active movement of salts out of the tubule from a place where the salts are highly concentrated to less so establishes the gradient that we see here. Again, we have high salt concentration here, some moves out. A little less salt here, some moves out. A little less here, out. A little less, out, and so forth, establishing the gradient that we can see from cortex to the medulla. And remember, water cannot pass the ascending limb, so that the interstitial fluid is simply less and less and less and less concentrated as you go up, or the way to think of it is more and more concentrated from cortex to the medulla. So let's go over to the descending limb and take a look at it. The filtrate becomes more and more and more concentrated in the descending limb of the loop of Henle because this section of the loop is impermeable to salts, but is permeable to water. So the salts remain within the tubule while the water exits by osmosis. Thus, the filtrate becomes highly concentrated with those salts here, and then as it makes its way up the ascending limb, those salts are moved out, more here than here, more here than here, more here than here, and so forth, establishing the gradient that you see. Now, the concentration gradient through the medulla, the interstitial fluid in the medulla, serves to draw water out of the collecting duct back into the bloodstream. This is osmoregulation, and the urine is very concentrated with urea as a metabolic waste. Once again, the descending limb of the loop of Henle is permeable to water but not salts, resulting in a very highly concentrated filtrate at the bottom of the loop of Henle. The filtrate moves into the ascending limb, and the ascending limb is impermeable to water, but that salts are passively and then actively moved out of the ascending limb. More here than here, more here than here, and so on. And this establishes the concentration gradient in the interstitial fluid seen here as a change in the pink coloration. Hypertonic in the medulla and hypotonic in the cortex. As the filtrate then flows into the collecting duct from the cortex through the medulla, water flows out of the filtrate back to the bloodstream. This is osmoregulation with the production of a very concentrated urine. Here is my simplified explanation in writing. The descending limb is permeable to water, but not salts. Water passes out from the descending limb, concentrating the filtrate, concentrating those salts, in the medulla, deep in the base of the loop of Henle. Sodium ions are pumped out from the ascending limb. The ascending limb is impermeable to water. Sodium is pumped out as the filtrate moves up the ascending limb of the loop of Henle. And the extracellular fluid, the interstitial fluid around the tubules, displays a gradient, hypertonic in the medulla, hypotonic in the cortex. The filtrate, as it enters the collecting duct, is relatively hypotonic, but as it passes down into the medulla, in the collecting duct, but down through the medulla, the water flows out back to the bloodstream, and the filtrate becomes more and more and more concentrated, and this filtrate will flow into the pelvis of the kidney on its way to become urine in the bladder. Here is a fun diagram to study. Ultrafiltration at the glomerulus as the filtrate flows into Bowman's capsule. Reabsorption and secretion along the proximal convoluted tubule. In the descending limb of the loop of Henle, the filtrate becomes concentrated as water flows out of the tubule into the interstitial fluid. The transport of salt out of the ascending limb establishes a gradient, hypertonic interstitial fluid in the medulla and hypotonic interstitial fluid in the cortex. then a little bit more reabsorption and secretion, and as the filtrate passes down through the collecting duct deeper and deeper into the medulla, more and more water exits to be reabsorbed by the blood. This is osmoregulation. Before I bring in the role of the hormone ADH in the production of a concentrated urine, let me digress to a different IB syllabus statement that fits nicely right here. Explain why the length of the loop of Henle would be positively correlated with the need for water conservation in animals. A long loop of Henle would establish a stronger, more profound concentration gradient in the interstitial fluid of the kidney through which the collecting duct must pass. The longer the loop of Henle, the more water conserved. A long loop of Henle would conserve more water as the stronger gradient would draw more water from the collecting duct passing through the medulla. In contrast, the shorter the loop of Henle, the less water conserved. Animals that live in very dry environments, deserts, are adapted to the local environmental condition by having relatively longer loops of Henle. Animals living in aquatic environments where water availability is high, thus water conservation is not critical to success in that local environment, would have relatively short loops of Henle. Once again, organisms living in environments where water is plentiful have little requirement for water conservation, thus the loops of Henle would be short. Organisms living in environments where water is scarce have a high requirement for water conservation, thus the loop of Henle would be long. This graph displays the relationship between the thickness of the medulla and body weight. And this relationship between medulla thickness and body weight is not what interests us here. What interests us is the comparison among the three curves for organisms adapted to three different environments: arid, mesic, and aquatic. Notice the curve for organisms living in arid environments. This curve shows that these organisms living in arid environments have a generally thicker medulla as compared to the medullas and kidneys of organisms adapted to mesic or aquatic environments. The thick medulla seen in organisms adapted to dry environments represents the long loops of Henle required to conserve water. Now let's take a look at the IB syllabus statement surrounding the role of ADH, a hormone released by the posterior pituitary. Explain the control of ADH secretion by negative feedback as part of osmoregulation. Include the release of ADH from the posterior pituitary under stimulus by osmoreceptors in the hypothalamus. ADH is a hormone released by the posterior pituitary located just below the base of the brain. ADH, released into the bloodstream from the posterior pituitary, targets the kidneys, specifically the collecting ducts of the kidney. The release of ADH into the bloodstream to target the collecting duct serves to increase the permeability of the collecting duct to water, to increase the water reabsorption, thus serving to conserve greater quantities of water. ADH results in a more concentrated urine. This is a diagram of the pituitary just below the base of the brain. The posterior pituitary is on the right of this image. At the base of the brain is the hypothalamus, and the hypothalamus monitors the water balance of the blood. The osmoreceptors in the hypothalamus monitor the blood osmotic potential. Now, if a person is dehydrated, where water conservation is important to homeostasis, ADH would be manufactured by neurosecretory cells in the hypothalamus that stretch down from the hypothalamus to the posterior pituitary, where the hormone ADH is released into the bloodstream to target the collecting ducts to conserve water. This is a diagram I've drawn to represent the homeostatic relationship of the posterior pituitary and the collecting ducts of the kidney in osmoregulation. Let's start with conditions of dehydration. ADH would be released by the posterior pituitary to target the collecting ducts of the kidney to conserve water, serving to raise the osmotic potential of the blood, causing the ADH to be inhibited. ADH would be inhibited, resulting in more water released in the urine. As water is released in the urine, blood osmotic potential would decrease, causing ADH to be secreted, resulting in water conservation by the kidney, etc., etc. In writing, ADH targets the collecting duct, increasing the permeability of the duct, which increases the amount of water reabsorbed to the bloodstream, reducing the amount of water lost in the urine. When water availability is high through inputs of eating or drinking, ADH secretion would be inhibited, resulting in greater water loss in the urine. If blood water potential is low, this is monitored by osmoreceptors in the hypothalamus. If blood water potential is low, you feel thirsty and you would be tempted to drink. As well, ADH is secreted, targeting the collecting duct. Both of these events results in an increase in blood water potential, an increase which would in turn be monitored by the hypothalamus and would inhibit ADH secretion and would prevent you from feeling thirsty. If the blood solute concentration is high, water potential of the blood is low, blood solute concentration is high, the hypothalamus monitors that increases your sense of thirst, and ADH is released from the posterior pituitary, targeting the collecting ducts of the kidney to decrease the rate of renal water loss so that more water is held in the blood and increases the water intake from drinking. Now, both of these events bring the set point down where there is a decrease in blood solute concentration that would result in an inhibition of ADH secretion and a less sensation of thirst. As we move toward the end of this movie, here are two IB syllabus statements. Compare the composition of the blood in the renal artery and renal vein. Explain the presence of glucose in the urine of untreated diabetic patients. You've seen this table before, showing the concentrations of various solutes in the various regions of the kidney. Arterial blood arriving to the kidney has a higher concentration of urea than the venous blood leaving the kidney. Urea has been removed, excreted to the urine. Urea becomes highly concentrated in the urine as water, the solvent, is returned to the blood. The kidney serves to regulate water balance and excrete metabolic wastes. Arterial blood arriving to the kidney has the same concentration of glucose as venous blood, with the exception of a small quantity of glucose that's been used due to cell respiration. All of the glucose that is in the filtrate is reabsorbed back to the blood so that no glucose should appear in the urine of a healthy person. Arterial blood has more salts than the venous blood because salts have been excreted in the urine. Proteins do not enter the filtrate, as you already know, but notice that the protein concentration has increased slightly as the blood moves through the kidney. The increase in protein concentration seen here results from water loss to the urine, not due to an increase in the absolute mass of proteins in the blood. See me in class if you have a question on this. Now, it's important for you to think about blood composition details that are not presented in this table. Venous blood will be deoxygenated relative to arterial blood. As well, venous blood will contain more carbon dioxide than arterial blood. In this slide, I've circled the important numbers for comparison. I'll let you do that work. It's important for you to remember that venous blood will be deoxygenated relative to arterial blood, that venous blood will contain more carbon dioxide than arterial blood. In a healthy person, the blood arriving to the kidney has 100 units of glucose, and the filtrate has 100 units of glucose, but urine has 0 glucose. All of the glucose that passes through glomerular filtration to become part of the filtrate is returned to the blood, except for a small quantity used up during cell respiration, accounting for the small drop in glucose in the venous blood leaving the kidney. Now, a diabetic has so much glucose in the blood that the filtrate levels are also high, too high for the active transport processes to reabsorb it all. Thus, glucose can be found in the urine of a diabetic. And that brings us to the last two IB syllabus statements of the movie. State that blood cells, glucose, proteins, and drugs are detected in urinary tests. State that kidney failure is treated by hemodialysis or kidney transplant. Urinalysis tests look for the presence of various substances. The presence of certain substances could indicate disease, such as diabetes or kidney failure. Urinalysis tests examine the urine for the presence of white blood cells, presence of proteins, presence of red blood cells, or glucose. Additionally, drugs, whether they be performance-enhancing drugs for athletes or dangerous recreational drugs, can be detected in urine tests. Urinalysis involves examining urine under a microscope, as well as using test strips and more rigorous biochemical analysis. The urine of a healthy person will have numerous solutes dissolved in the solvent water. Various kinds of tests are conducted on urine to determine if a person is healthy. The urine of a healthy person should not have proteins, glucose, or red blood cells. Chemical analysis of urine can also determine if a person has drugs in their bloodstream, for example, performance-enhancing drugs for athletes or dangerous recreational drugs. State that kidney failure is treated by hemodialysis or kidney transplant. Dialysis is a process by which blood is withdrawn from the body and moved through an apparatus that serves to remove certain wastes from the blood, such as urea, and then maintain other substances in the blood, such as glucose. The separation of substances in the blood occurs on the basis of their relative abilities to pass through a membrane. The wastes are removed while the cleansed blood is returned to the body. Dialysis works on the principles of diffusion of solutes and ultrafiltration of fluid across a semipermeable membrane. Blood flows by one side of a semipermeable membrane, and a special dialysis fluid flows on the opposite side. Smaller solutes and fluids pass through the membrane, but the membrane blocks the passage of larger substances, for example, red blood cells or large proteins. This replicates the filtering process that takes place in the kidneys at the glomerulus. Dialysis is used for those with a severe loss in kidney function, but dialysis is regarded as a holding measure or stopgap until a kidney transplant can be performed. Here you can see the removal of a kidney. Often a new kidney is transplanted, leaving the diseased kidneys in place. Kidney failure is treated by hemodialysis or kidney transplant. And that brings us to the end. IB Bio Animal Physiology, Topic 11, Part 8, Kidney Osmoregulation, C.